There's another one in February. Then there should be one in March and April.
Hold on, kids. Hi, we can see you. Yes, welcome. I like the Skyview image. Hold on, I'm gonna try and get my TVs to turn on for the people in the classroom here. So, and then we'll get started here in just a second. Hello, Spotsy. You guys act like you've never seen a TV.
Good to see you too.
Thank you. All right, my TVs are not coming on for whatever reason, so hang tight. Let me mute you and see why I cannot get my TVs on, okay? I'll be right back.
For study abroad. For a semester
Why
or
is it-
year.
Why it's.
I don't know why they have a Skyview. It's like, you gotta watch the football games and they have the. All right. Welcome, everybody. Welcome back, chemistry fans. I'm Mr. Garrison. I'm over here at Riverbend High School. Today, we have the pleasure of having a guest speaker. He is going to go until right up, I believe it is right around the 9:55, 10 o'clock mark. I will clarify that one here in just a second.
So, yeah.
But we're gonna save enough time at the very end of this, so stay in if you can. Dr. Grisham will answer any questions that you have. He will also share his email with you so that you can make some personal contact with him later on if there are considerations on your part from colleges, UVA specifically. What do they have to offer? And he knows some folks in there. Okay, so with that said, today we are going to have Dr. Charlie Grisham as our guest speaker. He is the associate chair at the in the chemistry department at University of Virginia. He got his undergraduate at the Illinois Institute of Technology. He got his graduate degree at the University of Minnesota. I believe we just talked today, we solidified 1975. He started at UVA. He is circling around at about 48 years of tenure there, at least. And I believe he said that Thomas Jefferson was not very good at racquetball whenever they were playing sports together, but he still claims that he beat. I don't think that this is true, but he says he still did. He did triumph over a quick game there. And so if there is something that you are wanting to pursue at UVA, he probably knows them. So please make contact with him. Introduce yourself. I know this is a virtual world, but he will meet you on the campus and he'll give you some personal touches, personal tours, and tailor it directly to what you want to study. So take this advantage to make contact with him. He is one of the nicest, gentlest individuals that you will ever meet. So please. Take the time. Now, I do want to share this with you also. Let me share a screen just to give you a little bit of context about how he is one of the premier in his field. He has recently, I believe, what was the copyright on this edition that I have, Dr. Grisham?
That one's probably about 10 years ago.
So it has gone on a while. Well, while you guys get to college, he is updating this currently. He said it is going to the editor as we speak. And so he wrote the book on biochemistry. He's going to love to tell you a story that whenever he started in this, there wasn't really much of a field to begin with. And then it has blossomed. And the stories that you're hearing in science-related news, he's helping develop some of that. He's created it. He has helped develop some of the individuals who are writing the stories. Okay, so without further delay on my part, I am going to now turn this over to Dr. Grisham, who is going to share with you a story about some bizarre biochemistry. So I would ask you to pay attention and ask any questions that are necessary along the way. He's receptive to the questions in the middle. If things aren't clear, ask away, and he will help you understand it a little bit better. I think I'm sharing my screen. Is everyone seeing it?
Yes.
Great. Okay, let's get started on this then. Dr. Grisham, maybe come in.
Happen to you and see what it's like. It might not be any good. Hi, everybody, and hi, everybody out there in the netherworld, wherever you are. It's really nice to be here. As Trent says, we've been doing this a long time, and it's always fun to come over here and do this. And I've met so many nice people, so many nice students. It's just been a treat all these years, and it's really been fun. I'm going to start out today, and I always try to tell something new in this story that Trent hasn't heard before, Mr. Garrison. And I came up with something that has a message for you too. I never told him that when I was in college, way back, that I had a TA, a graduate TA, helping me in analytical lab when I was a freshman in college. And he was just a graduate student. I didn't think much of it. Big, tall African-American guy. And he helped me with something that I was having trouble with. He was very nice to me. I'll tell you the story at some point if you want to talk. But the thing he did, I came into his dorm room one night and he had chemistry books all over the place, open to complicated equations I'd never seen before. And so on. I wasn't going to be a chemistry major at that time. I was going to be an engineer and build circuits and things like that. But I looked at what he was doing and I said, why are you doing this to yourself? It looks really hard. Because he's a graduate student of chemistry. And he gave me this big smile and he said, because chemistry teaches me how life works. And I never forgot that. And a couple of years later, I had this wonderful internship at a General Mills, Betty Crocker food research lab, and it hooked me on biochemistry, and here's I am today. So chemistry teaches you how life works could be the title of this chat, okay? This is what we're gonna do. So we're gonna step through this, and if I pick this, we'll go to slides, good, okay. So I'm gonna tell you a bunch of stories. The whole point of this is they're little stories that are connected with your life somehow, what people look like, or the color hair people have, and that sort of thing. And then we're going to show you a little bit of the chemistry that explains all of this, and then we're going to start a new story. So just enough chemistry to get in and show you that it's relevant, and then we'll move on to other stories, because I don't want to lose you and talk too deeply in one subject. So this one's about water. This one's a story about everybody drinks water. I got water up here on the podium. Water has been called the elixir of life because you can't have life without it. If you ever go to the Baltimore Aquarium, there's a big sign, big display at the front when you walk in, and it says that water is central to everything we do, essentially. Without water, there's no life, and with water, there can be life. So you'll see that if you go up there. So the story here is that Mark Twain even talked about water. He said whiskey was for drinking and water was for fighting over, because he'd been to the Southwest and he saw the terrible time that people had even then finding water. And now you know what's going on, that Lake Mead is drying up and there's going to be California water rationing and that sort of thing. Water is precious and we don't have enough of it. And climate change is making all that worse. So we'll tell you this story that life is bizarre, there's the title of the talk, in the desert and the ocean and in the Arctic. And you might not ever think that. You think, ocean, there's lots of water, right? The animals in there, a lot of them can't use it. Whales can't use water in the ocean, it's too salty. And so, and what about the Arctic? Well, polar bears in the Arctic have a really tough time with water in the Arctic. They're surrounded by water. It's too cold, either the seawater or the ice, and it's too salty. They can't drink it. They don't drink it. And so all these animals, camels, gerbils, Dorcas gazelles, and kangaroos in the desert, whales and dolphins in the ocean, polar bears do not drink water. They either don't have it available in the deserts or they can't drink it because of what it is. And so they all have a need to find water in a hostile environment. What do? Here's a picture of a polar bear. And behind the polar bear is a scientist by the name of Malcolm Ramsey. He was famous for studying individual polar bears for months at a time. And he would follow individual bears for 6, 7, 8 months. And he discovered that adult polar bears do not urinate for up to 8 months at a time. They can't afford to because that would lose water, and they have to retain their body water because they can't drink the seawater and they can't eat the ice. So they go long times without urinating, so there's not as much yellow snow in the Arctic as you might imagine. So the question is, why do you urinate? Why? It's not to get rid of water, it's to get rid of nitrogen, okay? You urinate in order to excrete nitrogen. You're eliminating excess nitrogen. We all get nitrogen from the protein we eat, okay? And other foods like fats don't have much nitrogen. So the polar bears turn out to only eat fat. Adult polar bears will catch seals as food, and they'll eat the blubber from the seal, but they won't eat the flippers. Because the flippers are muscle, and they don't eat those. So though they don't have to excrete nitrogen in the form of urea, you urinate a chemical called urea every day when you go to the bathroom. Polar bears don't consume much nitrogen, they just eat the fat from the seals, so they can afford to not urinate. Don't try this at home, it's not good for you. But it only eats fat, they only eat fat from the blubber of the seals. And this saves their body water because they don't have to urinate. Okay? And that's very efficient for them. So what do they do for water? They're going to run out of it, they're going to breathe it out. So how do they make water? They make water in the same way that your car does. And you don't think about that, right? You turn your car on in the winter, it's getting cold now, and it's a cold day, big clouds of stuff come out of the tailpipe. What is that? It's not CO2 because that's colorless. Water vapor in little droplets, clouds of water, because when you burn gasoline, you're also, it's just like burning fat in your body. Okay? So the polar bear and your car both turn fat into water. Fats and gasoline are full of hydrocarbons, carbon and hydrogen, right? And lots of electrons, lots of hydrogens, and oxidation of these molecules, CO2, and I know you've studied that already, requires that you put those electrons and hydrogen somewhere, and water is the result. And so what it turns out that 116 carbon fatty acid, long fatty tail on this molecule, plus oxygen will give you 16 CO2s and 130 waters when you count all of the byproducts of the metabolism of the fat eventually. But at the convenience store, we crazily enough spend $350 a barrel for water. You know, oil right now is about $100 a barrel. Water is $350 a barrel. Was that nuts or what? Buy it in little tiny bottles like this, and if you scaled it up to the size of a barrel, it'd be $350 a barrel in water, which is crazy. So here's a story about the polar bears from a cartoon from years ago, and your folks will remember these, the Far Side cartoons. And this shows the black bears going to visit the polar bears in the Arctic. And the guy that wrote this cartoon knows his chemistry and biochemistry, because the caption says, "Don't eat the flippers, Zeke, or they'll know we're tourists." So the polar bears don't eat the flippers, and that's because they can't consume nitrogen. So they retain all their body water, they make their own by burning fat, they never have to urinate. It's a great story. But then they live in the Arctic, and the question for you is, where'd the name Arctic come from? It's from the Greek, Arctikos, which means land of the great bear. But the Greeks never saw a polar bear, ever. Polar bears never made it down to Greece. Why the name? Why was the Arctic the land of the great bear? Anybody in here know? Anybody?
The constellation?
Yes, the constellation, the great bear, Ursa Major, the great bear. That's the Big Dipper. And you can see the tail of the bear is the handle of the dipper. Here's the dipper. And the rest of the bear is hard to see these days with light pollution, but it's up there. And in the northern hemisphere, which Greece is, you can see this constellation 24 hours a day if it's dark enough, all year round. It's always up there circling the North Star. So you can always see this constellation. So they saw a bear in the sky, and they called the Arctic the land of the great bear. Even they'd never been to the north, they called it that. Good story. And speaking of cold things in the Arctic, here's another thing. Pandas in China, out in the wild, like to roll in horse poop when it gets cold. What do you suppose that's for? Can you imagine rolling in horse poop and what you would smell like? Polar bears do it all, do it all the time. You may not have realized it, but they really do. There's a link here for something that we reflect on later. Mr. Garrison will have the slides. Here's a picture of a panda who has rolled in horse poop, coated himself with it. Why? Why would you do that? There are chemicals in the horse poop that keep the panda from feeling cold, if you can believe that. Here's a video. There it is. He's rubbing horse poop on himself, okay? Here's a video. Here's another one. There's rolling in the horse poop out on the road. Isn't that gross? Can you imagine doing that? It just looks terrible. Why do they do it? New research just the last couple of years. This is brand new stuff, a couple of years old. The pandas do this so they won't feel cold when the temperature goes down. Horse poop has two molecules that block the little channel proteins in your skin cells that make you feel cold. Okay, and here are the examples. They're called karyopolines, and only horse poop has them. And they block the channels in the bears in the panda skin that make them feel cold. All vertebrates, pandas, humans, penguins, everything, sense cold with a protein called TRPM8, TRPM8. It's a membrane protein. It helps neurons to communicate to have you sense the cold. There's no reason that you would even notice the cold if it wasn't for this protein in your nerve cells in your skin. It's an ion channel that lets calcium and sodium run across it. And the thing is that when you feel cold, the channels open and the sodiums and calciums run across the membrane and you say, geez, it's cold, you know, but not if the channel doesn't work. So if you can block the channel, you don't feel cold. So then the temperature drops, the channels open, you feel cold, bad. Here's what it looks like. This is the protein. This is looking down into the membrane, and there's the pore, there's the channel where the sodiums and calciums go through. This is from the side, it's a great big protein, and the channel goes up the middle like that. This is where the membrane is right here. And so when you feel cold, it's because this channel has opened because of a temperature change, and the ions cross, and you suddenly say, wow, it really feels cold. If you could block that channel, you wouldn't feel cold. So the bears rub karyopolines on them, and it blocks the channel. No more calciums flow, and they don't feel cold. And the cool thing about this, no pun intended, is, I just slipped that in, that penguins are different from pandas and different from you and me. Pandas walk around in the Arctic and the Antarctic, and they don't feel cold. They never sense the cold because their channel proteins a little different. The protein sequence and structure is a little different, and it has to get a lot colder in the Antarctic for the penguins to feel cold. So penguins can walk around all the time and not mind this. It's really interesting. So I say feeling cold is a state of mind. It's all in your mind. And if you could avoid the channel opening, then you wouldn't feel cold. Penguins happily live in the Antarctic and never feel cold because their set point for feeling cold is lower than the temperature of the Arctic. Isn't that cool? Again, no pun intended. Okay, next story. Next story. Why? Now think about it. You've all seen Renaissance paintings and Botticelli and that sort of thing. And why are there blonde people in Renaissance art? Because Italians in the time, in the Renaissance, were all dark haired. They didn't have blonde hair. Northern Europeans had dark hair. Had blonde hair rather. So why are there blonde people in Renaissance paintings? Everybody always wants to do what they can't have. It's exotic if you've got something from Europe or from around the world, right? Europeans want American genes and you get the idea. And so we have this interesting thing that you always want something, you know, the Italian women wanted to have blonde hair so they would look like this, and like this. So here's the story. You couldn't bleach your hair to do that. Hydrogen peroxide hadn't been invented yet, hadn't been discovered. Nobody had a source of hydrogen peroxide, and certainly not L'Oreal and things like this. But Italian women relied on plants to turn their hair blonde or lighter colored anyway, because plants have little tiny bodies in the plant cells called lysosomes that have hydrogen peroxide in them. So if you take a plant and you grind it up and you take that plant paste and you rub it on your hair and leave it for a couple hours, and then you sit out in the sun and you come back in and do this again and back and forth, rub the stuff in your head, go out, sit in the sun, back and forth and back and forth. eventually your hair lightens up. It's a lot of work, but now you've got exotic looking hair, right? Because there's a little peroxide in the plant that you can extract and use. Okay, so you get things like this, and people did this. And so the Renaissance artists all depicted the women as blonde-haired people, even though no Italian looked like that. So you get the idea. Here's another one, Venus, mythical being. Here's Lucretia. Lucretia was a real person. She lived 500 BC, and she was the daughter of a nobleman. And she was raped by the son of the king at the time. And she was so horrified by this that she committed suicide. She took a dagger and plunged it into her heart. This is supposed to be how the Roman Republic started. The Romans at the time were a monarchy, and the people in Rome were so upset that this had happened, and it was the fault of the son of the king. They overthrew the king, killed him, got rid of him, and supposedly formed the Roman Republic. We don't know if that's really true. It's a traditional story about how Rome was founded as a republic. Pretty good story. But anyway, how does it happen? Here's the peroxisomes and lysosomes in the plant cells that can lighten your hair, so you can end up looking like that instead of dark hair. Here's another story from Italy. It's completely different. Beautiful eyes. What are beautiful eyes? Well, at the time in Italy 500 years ago, it was imagined that the darker your eyes were, the more exotic you looked. So people tried to darken their eyes. You can't do much about your eye color, but you can dilate your pupils. And so people learned to use the plant called the deadly nightshade to put drops in their eyes and dilate their pupils. There's a molecule called atropine in the deadly nightshade that you can use to dilate your pupils. It's still used in eye exams, but Italian people, especially women of the time, dilated their pupils with atropine to make their eyes more beautiful. In fact, the name of the plant, Atropa belladonna, belladonna means beautiful woman in Italian because of this practice. And there were recipes at the time for what was called blonde water, aqua bionda, which was just this plant ground up and liquid extracted from it and then dropped into the eyes. Blonde, atropine dropped into your eyes would dilate your pupils and make you more exotic. And so people did that. Some people still use atropine today in eye exams. Most of the time you use something called tropicamide instead. That's what tropicamide structure looks like, but here's what happens. And the reason why you use tropicamide today instead of atropine is that it goes way faster. So if you just go for a routine exam, you're going to get tropicamide in your eyes. If you have to have a long eye exam, you'll use atropine, as it turns out. So that's important. So how does this work? What does atropine do? It works with another membrane protein. This is a membrane protein, and you can see these parts of the protein that are in the membrane here. And this is the membrane, and that's the outside, that's the inside. And it's called a receptor protein. It's a G protein coupled receptor, and it binds atropine to change its structure and change its function. And when it does that, you're going to get dilation of the pupils of the eye. So there's the receptor, there's a protein that interacts with it. What happens is that acetylcholine has to work with this receptor protein to control the size of your iris and how it dilates and so on. And so you get smooth muscle contraction that keeps the eye looking like normal. But if you can block that process with atropine or tropicamide and block it like this, then you block the smooth muscle contraction in the iris of your eye. And that causes the pupil to dilate. So atropine blocks the receptor and changes the shape of the iris in your eyes so that your eyes look darker. And why would people do this today? But that was the practice of it. So here's a connection between eye drops and mosquito bites, and another story that probably you wouldn't think about. We're going to take a more general picture of how hormones and odorant molecules and other small molecules act as biological signals. Okay, so let's take the same story, hormone, odorant, whatever, and a receptor protein like that, and all kinds of targets and signals result from that. Okay, so you get any kind of blocking molecule will change the action of a hormone and change the function down here. If you bind some kind of molecule to that receptor protein that changes its structure and changes its function, you're going to change the targets and the signals that result. That's what much of your signaling in your cells actually does. So we're going to talk about how you could confuse a mosquito. Okay? Nobody likes mosquitoes. Nobody likes mosquito bites. Mosquito repellents have something called DEET, and that's diethyl beta-toluene, DEET. And the question is, how does it work? For a long time, nobody knew how this mosquito repellent actually worked. A woman at Rockefeller University in New York, Corey Bartman, has actually figured it out. She studies worms, of all things, little roundworms that have little tiny brains and only with about 500 neurons. We have 10 billion neurons in our brains. These little worms only have about 500 neurons. And she may have an insight into how all this works, that she is. She studies these little worms that can't do anything but sense food, and she puts them in mazes and watches to see how they find their food, and then she maps this in their brain, because the brain of this little worm has been actually mapped, and she can figure out which food triggers which neurons in the brain, and she can actually study the function of the brain in these organisms. So how is this related to mosquitoes? Well, she's found one G protein coupled receptor, a G protein coupled receptor is a GPCR, in two sensory neurons in the mosquito, as it turns out, that respond to DEET. Okay, and DEET binding to this GPCR, as it's called, disrupts the connection between the odorant and the locomotion. This happens in the roundworm, it happens in the mosquito too. So think about how she discovered this with the roundworms. She gives them DEET, and all of a sudden they can't find their food. They're in a maze and they don't know where to go. The chemical stops their ability to find food. All they can do is use their nose. They don't have eyes, and they use their nose to find the food. If you block the action of the sensory molecules in the nose with this chemical, then they can't find their food. It turns out the same thing is true of mosquitoes. Here's what DEET or DEET looks like. And so DEET really isn't a repellent. It's just blocking the ability of the mosquito to find you. There's a lot of research that shows that the mosquito is finding you because of the carbon dioxide that you emit. And so Bartman calls DEET not a repellent, but a confusant. Okay, it confuses the mosquito, and so the mosquito can't smell you anymore, or may be able to smell you, but can't figure out how to get to you. Okay, so we think that DEET, the mosquito repellent, is really a confusant because it blocks these two receptor proteins that allow the mosquito to find you. Okay? Very interesting thing. It just confuses the mosquito. It doesn't really repel them. It doesn't push them away. It doesn't know where to go. Another story about blue people in Kentucky. And you'll like this. This is really strange. In 1820, a guy named Martin Fugate, a prisoner from Scotland, actually settled in a place called Troublesome Creek in Kentucky. And he had a blue cast to his skin. He had a medical problem that gave his skin a blue color. And 6 generations later, all kinds of other Fugates grew up, and they were all blue. Very interesting thing. And a doctor, a hematologist from Lexington, Kentucky, went to visit these people because he'd heard about this. He wanted to study it and see why it worked. And he suspected a condition called methemoglobinemia. I suppose that is. Here's a picture of some of the blue people at the time. Another couple of pictures. You can see some of the people with blue faces and so on. Here's a black and white picture, but this fellow on the right was known as blue ants for the color of his skin. So what's this condition called methemoglobinemia? Hemoglobin, you know, is the oxygen carrying molecule in your blood. That's why you have blood, is to carry oxygen to all your tissues so you can do normal respiration. And normally, the iron in the hemoglobin is iron 2+. We've all done oxidation states, right? You know a little bit about that. And so iron is iron 2+ in normal hemoglobin. But some of that iron in your hemoglobin is always oxidizing to iron 3+. It's giving up an electron and it's becoming iron 3+. And that hemoglobin is blue, quite literally. It goes from red to blue by turning the iron from 2+ to 3+. There's an enzyme that fixes this in your body. It's called diaphorase, and it repairs the hemoglobin by putting an electron back on the iron 2 plus and making it iron 2 plus again. So you don't know that this happens all the time to you. Right now, all of you listening to this are turning a little bit of your hemoglobin into methemoglobin with iron 3 plus, but your repair enzymes are putting it back to iron 2 plus so you don't look blue. That's the trick. The enzyme is saving you from turning blue, like literally. Right? And if the diaphrase enzyme is defective, then the iron 3 plus accumulates. So you can think of your diaphrase genes as blue genes. All right. So here's a little plot. Here's a little plot of percent methemoglobin unreduced. This is in someone who has a lot of methemoglobin. And a normal control is going to lower that right away and get rid of the methemoglobin. But if you were a blue person with 2 genes that are defective, if you're a BB homozygote, then you'll stay blue all the time. That's a problem. That's not a good thing. So Colleen, the doctor, decided he was going to fix this. And he thought if it's just oxidizing hemoglobin and the iron, you can fix that with a dye that has 2 oxidation states. So he used something called methylene blue, of all things. Okay? So we'll show you the story. It has 2 states. It's either blue or colorless, and that ties into how this works. So he took a patient that was blue, and he injected 100 milligrams of methylene blue dye into their blood. And within minutes, their skin was pink. Okay? So he gave methylene blue tablets to all the blue people. And as soon as they took the tablet, they lost the blue color in their skin. Their skin looked pink again. And they were delighted. They were thrilled. The other thing is, when your hemoglobin is iron 3 plus, you feel lethargic and tired. You don't have much energy because you don't get enough oxygen. Because methemoglobin does not carry oxygen well. So the people felt better and they looked better. So this was great, right? So it turns out that he hadn't really explained how the dye worked for people. And one old guy told the doctor, well, I can see the old blue running right out of my skin when he went to the bathroom, because here's how it worked, okay? Here's the reaction. There's the colorless dye up there, plus the blue hemoglobin, and they react together and they turn the hemoglobin red, but the dye turns blue. And what happens to the dye? You excrete it, you urinate it out. And so the hemoglobin turns from blue to pink, but the dye turns from colorless to blue, and you urinate that out. So the people that did this said, yeah, my skin was blue, and then I just peed it all out, right? Everything's good. It's not quite that way, but you get the idea. There's chemistry in how they fix this problem. It's a really good story. How are we doing on time?
One more probably, about 10.
Oh,
So we have enough time for questions.
so we're going to show you a wonderful story. We're going to skip all the trees and cows and snakes and the aspen, okay? And we're going to tell you a great story about a wonderful plant called the Venus flytrap. And this is a video, okay? So you can watch this in movies. And Charles Darwin called the Venus flytrap, the plant that opens and catches flies and goes like that. You've all seen this. It's native to Virginia, by the way, down in the swamps in southern Virginia. This is where it lives, typically. He called it the most wonderful plant in the world. The flytrap uses an incredible collection of biochemical molecules to make the trap able to hunt flies and to catch them and eat them. Okay? There's a segment on the Discovery Channel called the Life series. And if you ever are looking for a Christmas present, get your folks to get you a subscription to this, either the DVD or online. Here's a video about the Venus flytrap from this. And I want you to all think about who you're listening to, who's narrating this wonderful video of the Venus flytrap.
Venus flytrap.
Recognize the voice?
Like the sun, it makes itself very love, using fragrant nectar across the surface of each leaf. Eating's good here, but any visiting insect better watch out for these 6 tiny hairs. Fly doesn't know the game is rigged. If it strikes one of these hair triggers, it can carry on feeding. But that contact activates a 20-second timer. Second contact while the timer's running, and an electrical impulse is triggered and the leaf snaps shut in a fraction of a second.
That'd be the only time you feel sorry for a fly.
Locked together like prison bars. If the fly is very big or very small, it may just beat the wrap. But most get the death penalty. Minutes later, wrap reopens. That remains is a desiccated husk. The rat has finished its meal and once again sets the table for the next victim.
So we'll tell you that there are a whole arsenal of chemical tools that that trap uses to do that, okay? It's a whole bunch of chemistry that works together to make that trap shut in a fraction of a second. And there's parts that are called mechanosensing hair cells, hair cells that respond to touch, okay? Those little things that the fly was touching. When that thing tips, there's a protein down at the base that changes its structure. And again, ions cross a channel, and that causes part of the closure. There's also water in the hinge region, and water channels are changed to allow that to happen. And so you've got ions crossing membranes and you've got water crossing membranes. And in an instant, all of that causes the trap to shut. So we won't be able to tell you the whole story. I think we should probably save some time, but we'll tell you there are what are called acid pumps that pump protons across the membrane. There's water channel proteins. There's enzymes, even a diaphragm, believe it or not, remember that, to digest the dope, as it turns out. And there's all kinds of things that come together to make the trap close. And we now are starting to learn the structures of the molecules that do this. I'll show you some of it. Here's the mechanosensing channel proteins. This is at the base of those hairs that the fly was touching. This protein is deep in the cells in the base. And when the hair moves, that channel opens up and allows ions to cross and causes the closure. It's just incredible to think about. It's like nerve impulses that you use to communicate in your brain. Same sort of thing. The plant actually has nerve cells that communicate the hair movement to the place where the hinge is, causes it to close. And then there's proton pumps, acid pumps, as we mentioned, that are involved in all this. We're learning the structures of these molecules just in the last couple of years. So it's a great, great story, and we won't get to the Nobel story. But anyway, the point of all this is that in the recent years, in just the last 10 years, we've learned so much about this. I'll give you one example to close to show you how much the world is changing and what opportunities are there for you. When I started as a graduate student, there were 4 proteins whose structures were known: hemoglobin, myoglobin, an enzyme called carboxypeptidase, and an enzyme in your saliva called lysozyme. Only 4 proteins we knew anything about. Today, we're about to cross over the threshold of 200,000 protein structures that we know about, and they're all in a database. If you go to a database called rcsb.org, you'll see a little thing up at the corner that tells you how many are archived there. 200,000 approximately proteins are known. We've learned so much, and what this means is these are opportunities for all of you. You guys are going to go on and do science and that sort of thing, some big medicine, whatever. And all of the things we're learning now are going to affect your life in the way that you run your career someday, but also your ordinary life. You will benefit from new drugs, new therapies, and that sort of thing. And it's going to change your world and our world. And it's because the knowledge is increasing so fast and so incredibly fast. It's a wonderful world out there in terms of what we're learning. And that's your opportunity to go on and do research yourself, go into medicine, go into whatever it turns out to be, that we use all this knowledge and change the world. Thanks very much.
Thank you.
Let's go ahead and turn this back over to you guys. This is your opportunity to ask questions of Dr. Grisham. If there's anything out there that you want in the chemistry world, he's connected. He probably has done some research or read up on it, can answer those questions. If you have questions about UVA and you want to know something specific about that, this is your chance. Ask your questions. I know some of you may have to leave, and that's perfectly fine if you get off the broadcast. Let's share your email with them. Is it on the cover slide there by any chance? Or-
Oh, it's not there,
Okay, so
but it's real easy.
let's write this down. Look at that. That's right there. Bond Energies. All right, let's let Dr. Grisham, you write your email. So if someone wants to connect with you, they can. And then go ahead, fire on just one at a time, any of the sites. If you have questions or anyone here has a question, please go ahead and help yourself to questions. Otherwise, if you have to leave, I thank you very much for your time, folks.
North Stafford has a couple questions. Go
All right,
ahead.
go ahead.
Go ahead, Ayana. She's moving so you can hear her. Go ahead.
So at some point you mentioned I have to like be in front of the
Well,
camera?
no, but he can't see me. So you just need to hear you.
At some point you mentioned was it inhibitor cells that change the shape of the eye to make them darker? Is there any way this process can be undone to not make the eye dark?
That's a really good question. And the answer would be, yes, there actually is. I'm not sure it's practical in terms of therapeutics, but the original function of that receptor protein is to bind something called acetylcholine. That's a small molecule that you have in all your nerve cells, or at least most of them. And so if you could take the iris that had been inhibited by atropine or tropicamide, and you bathed it in acetylcholine, the acetylcholine would displace the atropine or the tropicamide, knock it off the protein, and regular function would resume. Does that make sense? So, and that gets to one of the really neat things about proteins and how they work. They have binding sites where molecules bind. And if one molecule binds, you can often knock it off with another one, and they work differently. One molecule may activate that protein, the other one might inhibit it. So we talk about competition between the right molecule and the wrong one. And in this context, the atropine or the tropicamide would be the wrong molecule binding, which it binds, but it freezes the protein and prevents it from functioning. But the acetylcholine can knock the tropicamide or atropine off and make it function again. So you get binding in both cases, but one binding is proper and positive, and the other one inhibits. Does that make sense?
She's nodding her head. Yes.
Good, okay, great, wonderful. So, and that's actually a very important thing that biochemists study all the time is competition for binding sites. And we've learned how to make drugs by finding molecules that bind very tightly together. Many of you have parents that take statin drugs, right? And statins are inhibitors of an enzyme because they bind tighter to the enzyme that makes cholesterol. One of the enzymes involved making cholesterol. It binds tighter than the regular molecules that need to bind to that enzyme to make cholesterol. So the statins bind really, really tightly. That's why you can take a little tiny pill and knock out cholesterol synthesis all through your body, because it doesn't take much of this statin molecule to knock out cholesterol synthesis. It binds so tightly that it competes with the right substrate, the right reactive molecule. Other questions? Other questions here?
So.
Go ahead. Yes, just project your voice so other sides can hear you.
So I want to go into a career in either chemistry or biology, nutrition, like.
There are wonderful options. There's so many things you can do. There are multiple different fields or subdivisions or subparts of chemistry as a science. One is synthetic chemistry, where you learn to make new molecules, make drugs, and synthesize molecules and so on. There's an incredible effort now on catalysis, because people are looking for ways to produce hydrogen in large quantities from other molecules. Take a hydrocarbon like oil and do a reaction with it, catalyze a reaction that releases pure hydrogen gas, H2. If you could do that, you've got unlimited energy, because hydrogen combines with oxygen to make water and releases a tremendous amount of energy. So there's energy research that involves catalysis of breaking up of hydrocarbons to make hydrogen gas, or saving the world in terms of climate change and everything. That's a wonderful field. Biological chemistry is a huge part. Probably half the people in our department right now have some kind of biological chemistry connection in the chemistry that they do. So they study proteins, they study lipid systems, the vaccines that how many people have taken COVID vaccines? Like a lot of people. Those vaccines use a special kind of lipid chemistry or fat chemistry that has been developed over the last 40 years. And because we knew how these little fat molecules, lipid structures work, we were able to build these vaccines very quickly. There's structural chemistry, where you use very powerful electron microscopes and techniques like nuclear magnetic resonance and mass spectrometry to study structures of molecules. It's very electronic and very physical, but you can do that. And so there's all kinds of different subfields, and it will depend on what kind of things you like to do. And the other part that you should know about is informatics, because chemistry is becoming very computational. The proteins you saw, remember I said there's about 200,000 proteins these structures have been determined. There's millions of proteins we know about. And the problem with proteins is we've never been able to figure out their structures without very complicated experiments with crystals that you make from them and so on. But now there's a piece of software developed by Google, believe it or not, subsidiary of Google, and it's called DeepMind, is the division of Google that does this. You could Google DeepMind and read about this. And they've developed a software suite called AlphaFold, which can predict the structures of nearly any protein. And it's changing the world in the way that we study proteins, because it can predict the structure of a protein, even if you don't know what it is. And that's very computational. People write software to do chemistry, if that makes any sense. And so if you like computers and chemistry, that's a subfield that is just exploding these days. It's growing and growing and growing. Does that help a little bit? Yeah, there's so much you can do.
So on AlphaFold, is it like actively used right now, or is it still?
Yes. Oh, it's so neat. Google is giving AlphaFold to the world. You can run it online, or you can install it on a server farm. We have a server farm essentially called the Radata cluster at UVA, and we've downloaded the software for free from Google and DeepMind. And my own students are using it now to predict structures of proteins. You submit a job with 2 files that you send in, and an hour later you get back this incredible thing. And it's compared your protein sequence with all the known structures that are out there, and said, based on what we know, studying millions of structures and thousands that have been documented, we can predict that your protein should look like this. It's just, you would not believe it. It's free. Anybody can use it. You could do this. Look up AlphaFold. You'll fall over it. It's one of the coolest pieces of software. It took years to develop. It was very expensive. It's free to the world. It's really incredible. Yeah.
What would you say is the hardest part of your research?
The hardest part of research is not giving. Anybody doing research takes on a new problem that no one's ever done before. When I was a graduate student, the problem I got was something that nobody had solved up to that point. Now it's trivial because it was done a while ago. But when I took it on, I didn't know what to do. We tried many, many different things. I spent a year just trying to purify the protein I was going to use. And it was so difficult to purify because it was a membrane protein. If you took the fat molecules, the lipids out of the membrane, the protein died. And the hard part was not giving up. I walked home every day from the lab and said that was a waste of time. Nothing worked. You know, it was a failure. But the people that don't give up are the ones that get the answers and figure out the ways to do things. So the hardest part about research, believe it or not, is in your head. It's if you've got to have the stamina mentally and the strength to say, I'm going to solve this problem. And every chemist, every biologist, every engineer who solved a problem will tell you that the way to succeed is just to say, I'm never going to give up on the solution. And if you've got that mindset to refuse to give up even after a year of failures or whatever, you're going to get it. You're going to solve problems. That's how all research is done. Everybody who's ever done research has had to learn that lesson. That's the hardest. If you can do that, you can do research. What do you think? Yeah. It's just confusing to do.
All right, Spotsy, do you have any questions left over for Dr. Grisham? Guys, okay?
I think we're good. Thank you very much. Thank you, Dr.
Thank you very much. All right, we will talk with you again soon. Merry Christmas. Bye-bye.